Hello, everyone. Welcome to today's session. Before we actually start with today's session, I would like to go back and revisit the organization page so that we could recall the progress report and till now what we have covered and what is expected in today's lesson. So I'm going to share my screen now. Okay. I hope everyone could see my screen. Yeah, Justin.
Sorry, that was an error.
Oh, okay, that's fine. This is our organization page. If you go to the bottom of the EDU site, there is a direct link for the organization page that says Bioinformatics Training. So that's how you could access the organization page. And we have mentioned all our updates over here, the files and the presentation that is to be used for today's session. All the questions that were asked in the previous session or whatever the recordings or the files were used in any of these sessions are being added to the forum, so you can still go ahead and access all these things. And if there are certain questions that needs to be answered, could be posted here on the forum. And if I move to the progress, I would like to request every participant to go through the online modules that we have on our edu site, so that you have a clear understanding about what's going on in the session, and you could have a more hands-on experience. This would help you later when you are going to start on your project. So that is why it's very essential to go back and look at all the online sessions. So till now, if I talk about the modules that we have covered on edu side, so we have gone through transcriptomics 1, 2 and 3, along with introduction to bioinformatics. That's the first lecture that we did. And whatever we are going to cover today, the supervised and the unsupervised machine learning, that is for today's session and the coming up session on the next week, could be covered by going through the machine learning module that we have over here. So kindly go back and make sure that you go through online modules and do some hands-on practice on the server. And as I was mentioning before, that you can track all the activities that you do on edu site or on server, and the points are assigned to each and every activity, and you could see your own personalized progress report over here under the tab of progress. uh, and I would like to cover one more last thing. So what all we have done till now, and what are we going to do in today's session. So we started up with processing of the NGS data. In that, we discussed about the role of pre-processing in the standard RNA-seq pipelines and what were the mapping techniques that you could possibly use. Then we moved forward to the exploratory data analysis, in which we covered about the filtering and how you could normalize the data set. And then in the next lecture we covered about the analysis of gene expression, like how can you do the correlation or the clustering of the samples using the gene expression profile. And in today's session we are going to talk about the supervised data mining, and how could you exactly find out the difference in the groups that you have recognized. So now I would like to have Mohit, who is an expert in machine learning and computational biology, and he would be giving us an overview of what is expected in today's session. Thank you.
Thank you, Sahil. Hello, everyone. Can you hear me, Sahil?
Yeah, we can hear you.
Yeah. Thank you. So let me start to share my screen first. Let me know if you can see my screen.
Yes, we can see your screen.
Wonderful. So yeah. So hello everyone. My name is Mohit Mazumdar. So before getting into the presentation, I did my post graduation in bioinformatics and PhD in structural bioinformatics from Jawaharlal Nehru University. So before I start, during my PhD, the question we were trying to address was a classification and a prediction problem. So I used machine learning algorithms, classifiers to solve the problem. Then I built predictive models and published the findings. So today I'm going to talk about the basics of some of these classification methods, especially the supervised machine learning methods that are available and have been widely used to analyze the biomedical data. So machine learning can be classified into two categories. One is unsupervised machine learning, and the other one is supervised machine learning. So earlier, we used methods such as PCA in the earlier classes that you have seen. The methods such as PCA to understand and classify samples based on the gene expression, which is an example of unsupervised machine learning method. So the second type of machine learning method is supervised machine learning method, and it's been widely used in pattern recognition problems for a lot of different kind of data sets. So going into the supervised machine learning method. So machine learning, as I said, the supervised machine learning method relies on availability of training data that has to be that is labeled. So when I say training data that is labeled, it's like any other data set, which any other data that has been published and for which we know that the outcome. So for supervised machine learning method, we need to have information before. So we have a data set for which we know in and out that this is the gene expression for this level of data. So to understand it further, machine learning method takes in data which is labeled typically by us humans. In biomedical projects, this could be clinical data, images, or phenotypic data. The labeled data is used to train a model. So once you have a labeled data, you use that labeled data or the knowledge or the information to train it to a model. So the model varies, as do the principles used to build the model by using different kind of machine learning methods. So different machine learning methods will use a different mathematical model, and it will generate that model based on that training data set. So moving ahead, so what we are trying to do here is to predict the class of source from the source data. So the source data, as you see in this example, you can see different type of shapes and object of different type of colors. So if we can classify them as labels, so we can classify them on the basis of color. So you can see there is like green color with two different shapes and red color with two different shapes, and henceforth. So this will build our training data set. So once we have a training data set, we will use machine learning methods such as linear LDA, linear discrimination analysis, random forest, or methods such as SVM, which are very much popular, that has a capability to understand the large high throughput data. So the label data, yeah. So as I said, once the training is established, once you have a model or a template that can be applied to new data sets, so the new data can be assigned to a class. So what are you trying to do here is to do predictive analysis based on a training model, based on a model built from a training data set. So in this case, for example, once you have a shape like a star, which is not present in this data set, this will not recognize this point, and it will be a false prediction because the training data set does not have such shapes. So talking about data and the data set is very important in machine learning algorithms. That I've learned from my personal experience as well, because a lot depends on the way you train the data that you use to train the model. So in this case, if you see that we have divided the training, we have divided the data set into two groups. One is training data, another is testing data. So For example, here our input will be a set of genes with known levels and with their expression value. For example, a gene in symbol 465, I'm giving you an example, which has a certain expression in a certain cell line. For example, it's a luminal cell type, and this has an expression value of 1.52. So this is one example of your training data where you know everything about the data. So once you train the data and then you test the data, so how the algorithm works is it will develop a classification model, draw a decision boundary or a classification boundary with which it can classify the data into different groups. So we will go into much details of this after this during this session, where we will look into the different methods that is being used to classify biomedical gene expression data, and also we will look into the practical aspects using the tbioinfo platform, that how we are going to achieve those things. So what are the different applications that we use, that is happening around the world? We have been hearing about the buzzword of machine learning, artificial intelligence, and deep learning, and all those things. So what is the application? So machine learning applications, for if you see the clinical aspect of it, for clinical purposes, especially in clinical research, machine learning can be used to extract information about disease mechanism from a huge number of expressed genes. This has a very high value. So it can be used to select treatment for patients based on their mutation variants. So it can be used to prognose an outcome and improve the prognosis using new data. Some of these basic research questions can be answered using some of these techniques, which has tremendous translational value in clinic or in industry. Many machine learning methods used by pharma, uh, by crunching large data, helps in biomarker discovery. So these biomarker discovery tools, this biomarker discovery helps in developing diagnostic tools. So other than that, it could be used for detection of toxicology based on already known data sets, and you can predict that this is the stage and this is the data, that is what would be the outcome. Similarly, a lot of machine learning methods are used to decipher the molecular mechanism of drug action in using target discovery. So this machine learning algorithms can be used for target identification to to the molecule. So it has a high value in drug repurposing and repositioning as well, because we are looking at all the data that has been published. So there is also use in NLP, which is natural process language, which uses with which we uses with which we use to crunch and see the published literature and what is already been there. So there's a lot of application of machine learning algorithms in both clinic and pharma and biotech industry. So before I conclude this, I would just like to tell you that although data generation can be performed routinely, but analysis and data integration method still require active research and standardization. So you have to use multiple methods to derive to a conclusion. The secondary use, so there's an example which I'll give you a small case study. So for a supervised machine learning problem, when we combine genetic data with clinical data, the outcome is much better. So the more data you have, the better you will have accuracy. The model will be, the accuracy of the model will depend on that. So in machine learning, the more the data, the better it is. So before we jump into the details and demonstration of methods, I would like to say thank you to you all, and I would give the stage to Ilya now to have the demonstration and detailed understanding of some of these methods. Thank you.
Thank you very much, Mohit and Sahil. Let me share it. So I think if you can start. Okay. Okay, so if everybody can see my screen, let me start this brief brief presentation. And, you know, just an overview of where we are in this process of this whole program. As Sahil mentioned, we started with an introduction. We learned how to process next generation sequencing. And last couple of sessions we spent on exploring the different aspects of data analysis. And now we're kind of getting into this area of data mining that will prepare us for validation and then interpretation of our data. So one of the things that I want to say is that in the 3 lessons that we have online, Transcriptomics 1, 2, and 3, all of these things are already covered, and it's going to be very valuable if between today's session and the next session next week, you can spend some time reviewing the materials that we have online to kind of understand several different practical aspects of what we're talking about here. Now, as you all know, one of the ways to identify a clear pattern in data is to look at gene expression levels and compare them between several different classes. So in fact, we already did this in previous lectures. And here's an example of an expression of a single gene where we have some basal samples, cloud and low samples, and luminal samples. And what we see clearly is that in luminal samples, which are luminal breast cancer samples, we see this high level of expression. And then in cloud and low and basal, we have a lower level of expression. And generally, what we can see is that the threshold could be drawn around maybe 4, and that will be a separating boundary between those 2 samples, right? So then we can have this clear rule. If a sample has an expression level greater than 4, let us consider that to be luminal. And if it has an expression level below 4, it can be either basal or cloud and low. The next rule that we can say here is that maybe if the expression level is below 1, then it is most definitely cloud and low, because in the basal, we have 4 samples that have an expression level higher than 1. So that is just by looking at the single gene, right? And I'm speaking to you as biologists because in the end, what we want to understand is the function of, let's say, this gene. And we know that if the mRNA of this gene is more abundant in a sample, we can understand that this function is more likely to be present, more likely to be activated in the sample that we're studying. And then if it's not at all, let's say it's below 1, then we can say, okay, that function almost is not present in the sample we're observing. And in the basal, maybe it is, right? So that's where we might need more clarification. So now when we look at this example, this is an example from the data set that we already shared a link with you. And I want to take a look at a few other examples. How can we understand these patterns? How can we biologically understand the patterns using what's called regression, using a dependency, a correlation between these numbers? So if you open Excel, or if you go to the files that we shared, in the files that we shared, there is this folder called unsupervised 7,000 genes. And there we have cell lines expression data marked, right? So as you remember, we used this in previous sessions, so you should already have all of this data. And this data looks in the following way. We have a number of genes. So these genes, there's around 7,000 genes. And then we have all of different breast cancer subtypes that are labeled for us. So we can just see, without focusing on the specific cell line or the specific sample, we see a number of replicates for each condition. And so if you look at this data set, you'll see, for example, normal-like, basal, luminal, etc. To simplify the problem, let us just take two types of samples. So I did here 3 replicates of normal-like, 3 replicates of cloud and low. To do this, all I did was I selected normal, 2 of these normals, then I took another normal-like, and then I took some cloud and low. And I copied them over, and I have this kind of a table now. Now in this table, you can see that we have the same type of information, right? So we have an expression level for this gene for 3 normal-like and for 3 cloud and low samples. How can we measure difference? If I want to measure difference, let's say, between this sample and this sample, which are the same, right? So I'm expecting the difference to be minimal. One of the ways I can do this is just divide the number one by the other, right? So I can, for example, divide this number by this number, and I will have sometimes a problem. Sometimes I will have a problem because I will have a 0. And of course, it's impossible to divide the 0 by anything or anything by 0. But we all know from basic mathematics that if I just divide 1 by 1, it will still be equal to 1, right? So I can add 1 to each one of my input values, and I will have an accurate representation of the difference, right? So you can see here C2, so I'm dividing this one by B2, and I'm adding 1 here, and this will give me what's called a fold change. Now, why are we measuring these in fold change? Why don't we just say some universal value like 6? Is it high? Is it low? The reason is because, of course, these samples are generated under very different conditions, sequenced by maybe different machines, different hands that sequence them. And so therefore, we don't have an absolute measurement for gene expression. So in this case, when we measure a difference, we typically rely on a fold change value. So now that we're looking at this fold change, we can multiply this or extend this for all of our genes, right? And as a result, we know that if the value is close to 1, there's almost no difference. Or if it's 1, there's no difference. If it's a little bit close to 1, there's a little bit of difference, right? The farther away from 1, the more the difference. So if I take and I filter this out, okay, and I want to get genes that are exactly 1, I can say equals 1, okay, and look, I have a number of these, right? So in fact, I can take these columns, select all of my samples, right? And now I will have, let's see how many genes. I have 479 genes that have an exactly the same expression level, a lot of them zeros. So let's take a look at how do we summarize this data. We summarize it by plotting. This will be one sample, one normal-like sample. This will be another normal-like sample. And what we see is a straight line, right? And this straight line tells us that there is a perfect correlation of 1 between these 2 samples. So these 2 samples, just based on these genes, are exactly the same. However, there are many other ones, right? So let's go back. Let's delete our filter. Okay, and let's see, is that true for all of our values? Okay, so we see that no, in fact, there is a straight line here in between, and overall there is a trend that says that they are correlated, but at the same time, there's also noise, right? And so whatever doesn't fit the straight line actually means that they're not correlated. If I take this dot is not representative of this relationship. And in such a way, I can explore relationships between one sample and the other. right? The closer it is to being aligned to this straight regression line that says that they are the same, the better is the correlation, the more correlated they are. Now, I can also measure correlation just by using this function called correl, but here I need to use an array. So I can take all of sample 1, then I can take all of sample 2, and now I have 2 arrays, and I can see that the correlation between 2 arrays is going to be 0.88. The closer to 1, the more positively correlated they are, and you can see that comparing different couple of samples, I get different numbers, right? So now obviously I have 2 groups of samples. I have normal-like and cloud and low. The fact that these 2 normal ones have a fairly comparable expression values for my genes is expected. But what about cloud and low? So if I just take these 2 and I plot all of my gene expression, surprisingly, I also see that there are quite a number of genes here that will have the same line, right? I can see the line here, I can see the line here. So does that tell me that these 2 are also the same? The answer is, of course, no. Just having this line means that, for example, these 2 samples are living samples. They are both cell lines, they're both human cell lines, they're the same genes. So we do expect a lot of the samples, a lot of the data to be related. What
I'm really sorry to interrupt. Hello, I just have a question. Like, can you like use like 2 different colors, like for the normal and clouding, because it would be like easier, like helpful for us to see the difference really? Will it be possible?
Yeah, okay, so let me explain. These are actually genes. Each dot here represents a gene. That's why you cannot use 2 different colors, because what we are saying is that each dot is a gene. So there are many, many genes. There are 7,000 genes. What are different samples here are the axes. This axis is the first sample. This axis is the second sample. See what I'm saying?
Yes, 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 yes.
So therefore, it's not possible to color the dots in different colors, because the dots, it's not like genes. You see, each gene has 2 numbers. One number is from one sample or from one group of, one normal-like, and another one from cloud and low. But that, if I have to color them different colors, each dot will be a different color, because all of them are different genes. Do you see what I'm saying?
And I also have one more question. When we do the processing or like pre-processing and normalizing the data, we normally like remove the genes that are not expressed or which has like the values of 0. And here it's not possible like to remove the things like the values with 0 and do the analysis, because it will make us simple, right?
Okay, so it's not, when we do removal of 0, we are taking all of our samples, and whenever we see a 0 across all of the samples, we remove it. But if there is a sample that has some kind of a value, we can't remove it, because that's a signal. Okay, so the purpose of removing zeros from all samples is to eliminate non-informative genes. But whenever there is some value, they are informative. Just in this case, what we're doing is we are selecting only 2 samples from all of the available samples. So that type of filtering does not apply specifically here.
Okay, and one more question. I'm sorry really to interrupt, because these are like the biological replicates, right? For the normal and the clouding low?
Yes.
And can we take an average of all the 3 replicates of normal and the average of the clouding low and then plot the graph, or else like remove the values that has 0? And then, because when you have something like, for example, in the normal, you have some kind of like one replicate has a 0 and the other 2 replicates have some value. So in that way, we can we do in this way, like take an average and then redo the plotting everything?
I am getting to that point. So let me maybe continue with the presentation. I will get to address specifically, that's the type of an issue I'm getting to, okay?
Okay.
All right. So the idea here, what I wanted to show with these graphs is that we can study a relationship between a couple of samples. And the relationship is based on correlation, or based on a line that we can draw between these two samples. Okay, so as properly noted, what we can do is, for example, use t-test, right? And we've done that before. That will calculate not the difference between individual samples, right? So right here, right here, we calculate a difference between this one and this one by dividing them. That's called fold change. Or we can actually take the average between these three, and now we will find kind of the difference between the condition, right? And so I believe we already did this, but just to kind of go over, this is called t-test, and t-test comes from a test that was developed to take a sample from each group and see whether there is a statistical significance of the change between a number of replicates that we can define as a difference between conditions, right? So here, for example, we can do a t-test where we will take the 3 normal like, and then we will take the 3 cloud and low, and we will then get a p-value of the difference. And of course, the closer the p-value is to 1 means that there is no difference. The closer it is to 0 means that there is a difference. So we can repeat the same procedure for all of them, and then we can use the t-test again to filter, and we can, for example, get all of the ones that are below, let's say, 0.05. So let's select 0.05 as our cutoff, and now we will get those that are differentially expressed, right? So now we are looking for genes that are differentially expressed, okay? But however, the point that I was trying to make with this linear relationship between two samples is that this is a basic pattern, right? This line is a pattern. For example, if we select genes that do not have a pattern, let's do that really quickly. So let's do between, and we'll say it is between 0 and 0.4. 0.4, or let's say 0.7. Okay, so now we have a number of genes, genes again, right, that are not correlated between these two samples. So let's just quickly take a look at what would that look like. Okay, you can see that here they're spread out right here. They're not aligned in a single line, but 0.4 is still not 0, right? So there's still some linearity here, but the main point is that this pattern is not clearly defined. And so because this pattern is not clearly defined, and this pattern, a line, is clearly defined, right? That is basically how a machine, an algorithm, can learn structure of this data, right? It can take a look and it can say, if I do this kind of difference between all of my normal likes, there is a large component that looks like this, which means that they are related. They are in a single group. And then there are maybe other samples that look more like this, and that means that they're not related well. And so then I can group them in a different group, right? And so last time when we met, we talked about how using clustering, for example, we can start seeing those patterns in our data. However, the problem with this type of an approach, with a standard regression approach, is that specifically in gene expression data, we have multiple types of patterns, right? We talked about positively correlated or negatively correlated, for example. Positively correlated, we can basically represent two samples that have exactly the same or a very similar expression pattern, which means that if one is 1 and the other one is 2, one is 2, the other one is 3, if there's always a difference of 1 or some defined number between the expression values, this line might change the height, but it's still going to be a line, right? So we're looking for this line, and it could have a different slope, right? Now we're defining regression. It has different slope, right? Different angles. It has a different height, and this can be added to our regression formula. But it is a line, and that means that there is correlation. However, many times the relationship in high dimensional data will have these types of shapes, these types of patterns. And these patterns are not easily defined using standard linear regression. So the problem that we are facing is that there are going to be a lot of relationships that are non-linear in our data. And that is why we use machine learning, right? So we already talked about these being big and very different types of data sets. There are complex relationships, many unknown features that we don't know a lot about, and many features are correlated, but in a very different type of a way than we expect. And the main thing is that we see this interest in machine learning after the Human Genome Project was published, which means that a lot of this kind of data has become available. So quickly, this is something that Mohit will be talking a lot more in depth about in our next session, but I quickly want to go through some of these basic things so that we see how practically they can be applied to detect these types of patterns. As Mohit already mentioned, there are two main categories, unsupervised and supervised data. And for supervised data, we have to label them. As we saw in our example, we were basing our tests, for example, the t-test or some kind of an identification of correlation based on a very specific definition that these are normal-like and these are cloud and low, right? We knew that when we started it. So that is a requirement. And as Mohit was showing, when we have a sample with a bunch of different types of data, source data that kind of combines many different shapes, we can use some kind of a machine or an algorithm, which is the same thing, that says that we can define the relationship in a formula, and now this formula will separate the data into separate groups. And the way that these separate groups are related, right, between the samples within each group has some kind of a pattern. And we just looked what a pattern might be. So we saw a line as an example of a pattern. We can identify a more complex pattern that is going to, for example, in the case of this simple data set, represent different shapes of objects that we have within each group. Now, today, we're going to talk about specifically classification, but both classification and data mining are both a part of machine learning. And we are going to just first review how we can just define those shapes. And then we will essentially use the same idea of variance, the same idea of correlation, the same idea of relationship to build a classifier that we can then use for very specific purposes. So last time, or maybe a couple of sessions ago, we tried PCA, and so I want to just remind you what we did last in that session. We talked about, for example, PCA just based on variance, based on the differences between genes in samples. We produced a picture like that gave us a separation into two large groups. Okay, so let's quickly try this together. Let's go onto the platform. So we'll go to server. And remember, this is a data set that contains 4 different groups, right? So let us go into right here, unsupervised analysis under data mining. Remember to log in. Okay, and here I'm going to do it. You're welcome to do it together with me. All you need to do, let's go into the folder that you had. You should have downloaded this on your computer and had these folders unzipped. So here you can see unsupervised 7,000 genes, and you have this cell lines expression data. We will load it, continue, and run the PCA R library, right? So we are going to look at variance within each group and see how many groups kind of visually we can identify in the result. No need to change anything. Click on save, and we'll call this PCA. And let's run this pipeline. So now under my pipelines, I can see that it has started. So let's quickly, before it's done, talk about what are we looking at here. We're looking at a representation of samples. Each dot here is going to be a sample, not a gene. Okay, and this is just important to remember. We were just looking at the relationship between genes in 2 samples. Now we're looking at the relationship between samples based on their gene expression. So in this case, just visually, we can see that on PC1, which is this axis, right? You can see PC1, this axis X, so to say. It shows us a clear separation, right? We can draw a line. That means a clear separation is we can draw a line or a boundary between this group and this group, right? So let's see if we have a result. And please let me know now if you want me to repeat the process of just preparing that pipeline. Just want to make sure that everybody can do it together. Okay, so if there are no questions or any questions, so right now, again, what we want to make sure, yeah, let's repeat it together again. So Voki is asking to do that. So let's just quickly do that. Areas of analysis. And we want to go into unsupervised analysis. Okay, so if you click right here on unsupervised analysis under data integration and modeling. Okay, and here we need to upload the gene expression. So we're going to upload, add files, and inside the folder that I shared with you, there's an internal folder called unsupervised 7,000 genes. So we're going to take cell lines expression data, choose, has to upload, and then you press on continue, start. We can use either one. This will provide us with a little bit more options than this one, but for now we can just use the PCA R library. And here you can see that we have several things that we can determine. Number of principal components, whether we want to scale our axes, center them, and whether we want to transpose the table. Transposing the table will make samples as the ones that we are analyzing. And we are indeed analyzing samples, not individual genes. So we just do this and save. And now we can save our pipeline. So I'm not going to run this pipeline. because I just ran it, and you should also be close to getting those results. So let's just take a look at the results.
Just a question.
Here you can
in the PCs, like number of PCA components that you can like create, it's like 3, it's like standard, or you can create like number of something. And how can we choose like which kind of like PCA components we need to choose, like which is the best optional one?
I will show you the result and we'll understand the significance of that number 3. So let's just take a look at that really quickly, okay?
Okay.
All right, so you should get, you see PCA plots, and if you scroll down, it has 3 different images, right? Because we selected 3. So what did 3 mean? 3 meant that we will take PC1 versus PC2. PC1 shows the most variability. PC2 is the second largest, right? So you can see that this is 18%, this is 12%. Now we will see PC1 versus PC3, and you can see that PC3 has less variance, explains less variance. And we will also have PC3 and PC2. So that will happen if we choose 3. If we choose 4, it will do PC1 versus PC2, PC1 versus PC3, PC1 versus PC4, etc. All the combinations between those 4 principal components. Okay, so this is just to build this image. So what do we see in this image right here? We see that we have 2 fairly well-defined groups, right? We can draw a line on the PC1 component, and we will get a separation into 2 large groups. That is the point of PCA. Visually, it tells us the variance, kind of how we can group samples based on variance. Okay, so now that we've done this, we understand that this is really a relationship between different genes, right? So here, I just have an example for each one of your dots, okay? You can take some gene, okay? Here you have 2 genes, and you can see the profiles, those patterns. And you can see that the patterns are going to be non-linear. There's not a clearly defined, one is low, one is high. You have multiple relationships. For example, here you can see basal is kind of low, cloud and low is kind of low, but one is high. Then some of these go up and down, right? So the relationships are complex. And you can kind of play around with different genes and understand the idea of patterns. But using those patterns, we can now separate these into defined groups, right? So we already did this one time, so let's do it quickly together again. We can take this idea of variance, kind of like a broad definition of how these can be grouped together, and define it. And say, instead of doing everything that we can know, define 4, right? For example, we can use this number of 4 because we kind of expect this number 4, right? And we can say, make me a clear definition of 4 groups, okay? And what will this do? This will just produce a relationship between samples and select a certain threshold, a cutoff, where we can get to this number 4, okay? So let's quickly do this and we'll understand what that means. Again, we will go under unsupervised analysis. We're still in the unsupervised analysis. Okay, and here we will upload the files. Again, we can take the same data set, expression data, continue. And instead of PCA, we, for example, can use hierarchical clustering, or you can try any other ones that you want to do. Hierarchical clustering is easier to explain because we also get a dendrogram, and the dendrogram tells us what the relationships are, how many groups from the bottom till the very top, from having all of them to having 1 cluster, right? And here we have to remember that we do need to transpose our data because we're still looking at samples and not at genes. For distance, we can keep it at Euclidean, but you have several options. And here, for this specific data, WARD-D2 is a method that will work better. So we can just select that, but you can play around and try other ones as well and see what happens. And now we will just do hierarchical clustering and run this pipeline. Okay, now the idea is exactly the same. We will use some pattern in our data to define what are groups, well-defined groups. But the well-defined is going to be something that we will see improved after we reduce the number of genes, the number of features. Okay, so I just did this, and we'll see what the result is. Do I need to repeat this pipeline just so you can follow along?
Sorry, in the meanwhile, I have a question by the time the pipeline is running. When you set the parameters, like for the PCA, for example, like you set it at for 3, and what is like the final outcome, like on what basis we have to choose or something? So the highest variance is the best method or the lowest one? PCA 1 versus PCA 3?
Right, so let's take a look. Here's the PCA table. Okay, and here you will understand what the PCs actually mean. So the PCA table, instead of giving you a visual, gives you the source table from where the visual is being produced. So here you can see PC1 has 18%, PC2 is 12, 6, 4, etc. And you have a bunch of these PCs. In our next session, Mohit will talk a little bit more about what these PCs actually mean, but you can see that I can select any two for X and for Y coordinates and get a scatter plot of my samples, right? So here's what we saw in the output. We had one group, another group, right? Now, what if I change, right? So if I select data and I change this, and for example, instead of PC1 and PC2, I use PC2 and PC3. Okay, so now this picture changes. It's not as good of a separation, right? But I know the difference between this table or this picture and the previous picture. The previous picture explained in total of 18 plus 12%. So that means 20, 30 point something, let's say 31%. This picture explains less, 12 plus 6, so 18% or maybe 19%. So this is less of a good representation of what my whole data set shows, but nevertheless, it's still informative because it still tells me about 20, 25% of my data, right? So those are the meanings of these PCs, but we'll talk a little more technically in our next session about, you know, how do we arrive at which one is PC1, which one is PC2, etc. The parameters that you have, that number 3 that we put in, is just to tell us how many plots to generate, how many of these scatter plots to generate. You can always download the table, and you can see that it goes to, you know, PC52, or PC51 is actually the last, you know, reasonable one. So that is the output. Let's, for the sake of time, look at the results of clustering so that we can relate PCA to this result of clustering, okay? All right, so here again, we have a PDF, right? We also have a full table, we have the expression data, we have a bunch of other things, but let's take a look at the PDF. And in the PDF, we will see a dendrogram. Now this dendrogram, if we want to know the 4 clusters, what we need to do is we need to insert a line that breaks this tree, dendrogram, into 1, 2, 3, 4, right? So these are going to be our 4 clusters. And now we can clearly identify them. So we can say, okay, so this line represents the cutoff. Then here's our cluster 1. Here's our cluster 2. Here's our cluster 3. And here's our cluster 4. Okay, why did I say that these are these clusters? Because this line right here, right, it connects all of these sublines, and I know that it grouped them into this cluster. But I can also see that this is not necessarily the best way to do it, right? Because I can place this line lower, for example, here, and now we'll get 1, 2, 3, or 1, 2, 3, 4, 5, 6, 7, 8. 8 clusters. So now it's a question of interpretation. Do I want to do a cutoff at 8? Do I want to do a cutoff at 4? Do I want to cut off at 2, for example? You can see that I can move this. Let's create a new one. I can move it here, and that will give me 2 clusters, right? So this is something that I can put in as a user and get the appropriate result. But if we look at other results here that are not just the visual dendrogram, I can take a look, for example, in this table. It will provide me with the definition, a cluster number for each one of the samples, right? So you can see that objects are my samples. I have a cluster assigned to each one, and then the expression for that one. Okay, so these are going to be all of my genes. So based on some metric in this space, I can clearly define these groups. Okay, now we already did all of this in one of our previous sessions, and all of these things are defined in much more detail in the Transcriptomics 3 course. Okay, so please try and go through this to understand in a practical example how this could be used, right? And what I want to get to, right, this is, by the way, an example of clustering and PCA based on the 15 genes, not the 7,000 genes, but the 15 genes. You can see that we can get a very nice separation into the 4 clusters: luminal, cloud and low, normal-like, and basal, as well as here we have luminal, normal-like, basal, and cloud and low. So we can have an improved separation with pretty good explanation of variance. We can see 53 and 18%, right? So about 70%. That will show us a good picture of our data. But that is because we were able to somehow select some kind of a subset of features that is useful. As you remember from the first exercise we did in Excel, right, we can use filtering just to set some kind of a threshold on different statistical characteristics of our data to select some kind of a feature, select some kind of a gene, right? And selecting 15 genes by their pattern gave us a very nice separation. Now, typically, we use this information how? In biological interpretation, what is useful for us to do is to understand the relationship between gene expression, right? So we have gene expression, low is red, high is green, and medium is black, where we can now visually see these patterns. How do they work? Okay, and we can use clustering. So now you understand the dendrogram, right? That is the clustering, the reordering of these samples based on some kind of grouping, or genes based on some kind of grouping. And we can see very clearly defined patterns. Okay, they're not linear patterns, they're not just blocks, but we can see some patterns. So we can see that, okay, majority of these genes are highly expressed, right? So these, for example, 3 or 4 have a, or sorry, they're a pretty low expression, right? Here we can see a group of highly expressed. And now we can see the differences between different types based on this expression pattern, right? So what I want to get to is that there's a relationship between what we know about samples, what we know about genes, and these relationships, if we had to do this manually, would be fairly hard to find. But if we can prepare an algorithm or a model, a mathematical model that explains these relationships, now we can start using classification, because what we want to do now is to say, what if I have a bunch of data sets that I know something about, and then I want to take a new data, a new sample, just generate my transcriptomic data, do RNA-seq, and then predict where would this sample fall in my known structure of data, in my known pattern, my library of patterns. Okay, so this is called classification. And the easiest way to think about this is if I can select a gene where the pattern of expression is so clear that I can use a simple threshold and say, okay, based on this one threshold, just like here, you can see this gene, like we started in the beginning talking about the same gene. The pattern, if it's more than 4.25, assign it to this group. If it is less than 5.2, assign it to either one of these groups. Okay, that is really the point of classification. So before we do this practically, do you have any questions? Okay, as you can see, the other benefit of this type of an approach is it helps us select these genes. Not only can I classify that these are indeed basal or cloud and low versus luminal, etc., I now know based on which gene. And this is the gene that you see its pattern of expression right here, NSG, whatever, 116299. And this is another gene, as you can see right here, that gives us the separation into other ones. Now, the difference between these two approaches is that in the first one, we didn't have to know really anything about our data, right? We just put in a table and, okay, per team. So as you can see, just the question per team posed here, are these 4.25 or 5.25 random values, right? So why did we select these values? No, they're not random. They're based on the difference between expression, right? So you can see that the expression for all the low expressed genes reach almost to 4. And all the highly expressed genes are all above 4.25, right? You can see this one right here, that's 4.25. So everything above 425 is definitely luminal. Everything below 425 can be either basal, cloud and low, or normal-like, right? Because all the rest of them are below. Now, let's forget about these ones. We already know how to separate them, the luminal. And let's just leave it with basal and cloud and low. Let's find the lowest value, 5.2, right here, and say that everything above 5.2 is going to be basal. Everything below 5.2 is going to be the rest. Okay, so that is how these were selected, these specific values. But who selected them? We didn't select them. A person didn't go through all the genes and see, oh, okay, how can I separate it? It was a machine. It was an algorithm. So let's take a look at how that can be done. Let us go to supervised analysis. Okay, and here you can see quite a number of different types of analyses, discriminant analyses, classification analyses, etc. What they're built to do is build a model that provides a boundary, and then that boundary can separate effectively the samples in any given data set. You give it a new data set, if those patterns exist, it will be able to separate them. But here we need to provide two samples or two data inputs. Okay, here we need to provide at least two. Let's go to supervised, 15 genes. And here you can see test and train. What is the difference between them? The difference is that in the test, there are no labels. And in the train, we have the ID of the sample, and then we have a class, which is our label for that sample. So we have samples and then labels. Now you select both of them, upload the test and the train, and then you separate. So you can put the training under the training group, the test under the test group, continue. And now we can use, let's use decision trees. So decision trees right here, one of the simpler ones. There are no inputs needed here. I click on end. And run. Okay, any questions about? So if it wasn't very clear per team, it's because I was showing the result, right, before we actually generate it. But actually, this is a fairly intuitive idea. If we go back, right, it's what I was showing actually in the beginning. Okay, Nathan, which data set was I using? Just one second. The data set, and how did I separate? Okay, let me explain. So let's go back. We have all of these supervised and supervised, etc. Okay, so let's say we have this data set. Okay, and I'll show you how to prepare the test and train. Probably already have it open here somewhere. Okay, remember in my data set, I have some labels, right? I have known types. So here I have normal-like, basal, etc. Essentially, this is not an ID of this sample. This is a class. Okay, so if I know an ID, let's for now just say it's a number. Okay, I have, sorry, let me do this like this. So if I have some kind of a number as an ID, I have 51 samples, right? And for each sample, I know a label is a class. A label is some kind of, my definition of what that is. So this I can use as a training data set. Now I can tell the machine that this is what it needs to know about luminal, or all of the different data types I gave it for luminal, right? And based on this pattern, it can tell me whether, for example, sample number 44 is luminal or not, okay? And so forth. For each class, I gave it a number of representatives, and based on what I told it about the representatives, these values, it can now define any new data set that I give it, what it actually is. That's what I put in the test data. I didn't give it the labels. I just gave it some numbers. Okay, let's take a look. Yeah, this was just an example of how I prepared, but in reality, what I used was supervised 15 genes. So you can see here what the train and the test actually include. So let's open, first of all, the training. Okay, so let's open it up. Okay, you see the ID for each sample and the class, right? And how many are there? There's a bunch of them, right? So a bunch of examples for the training. Let's look at the test. Okay, I have much less testing ones, and they don't have a class. And so what I'm telling it is the algorithm get trained on this data, and then apply your knowledge, your information, your learning to this data, and tell me which ones they are. And now we'll take a look at how did it separate these samples and based on which genes. Does that make sense, Nathan? All right, so let's take a look at the result, and we'll see where did those numbers come from, right, to answer per team's question. Okay, so let's take a look at the output. And you can see here that we have all of the results zipped in the zip file. Okay, we unzip the file, and we'll have several different files in here. So let's first open the tree plot. Okay, you can see that we got these results. So for this gene, if it is less than 4.25, I can separate it. This is one group, and this is another group. And based on this gene, I can further clearly separate this one from this one. Okay, now let's see where did these numbers come from. Let us take a look at the original data set. Okay, so we can go, for example, unsupervised 7,000 genes, or sorry, unsupervised 7,000 genes. And here we have this marked file, right? So what it's saying is, let us find, okay, this gene, NSG. Okay, let us find this gene. That's what it did for us, is it actually found this gene for us. Okay, there's this gene. So let's copy it. Okay, we'll place it right here. And now let's go back up to the beginning and we'll just get all of our IDs. Okay, and now we can see what the pattern is. Okay, so what is the pattern? What it is telling us, right? What is this telling us? If it is less than 4.25, it's these two. If it is more than 4.25, it's this one. So let's find 4.25. It's right there, right? So this is 4, this is 5, so right in between here. Okay, we have, okay, 451 is the lowest one. So everything above this, why above? Because we have this guy right here at 351, right? And definitely is not a luminal, but you can see all the luminal ones. Okay, so let's just select all the luminal ones. Okay, where are the luminal? Here's a luminal, here's a luminal, right? Where else is a luminal? Here's a luminal, here's a luminal. No, this is just an expression, level of expression. That's all it is. The y-axis, these numbers I'm selecting are the y-axis. That is just the level of expression of this specific gene. Okay. Don't need this one. Okay, what we'll do is this actually has to go up. Okay. Something here doesn't go well. Okay, well, kind of messed up here on this one, but let's just select a few. Select a few. I clicked there on the wrong one. Okay. Okay, so here for you can see all the luminal ones are above that threshold. No, it has nothing to do with median. We are looking at all of our samples, analyzing the patterns, and the pattern tells us that all of them. Okay, if a median means that some could be up, some could be down, but in this case, it's all of them. All of the ones belonging to luminal will actually be above that threshold. All of ones that are below will be something else, right? And now we can see what is something else. So if we now select this gene, okay, let's find this gene in our original table. So right here. Instead of finding the first gene, we're going to find the second gene. Okay, and let's just select this gene and add it right here. Okay, and that is our second gene. Let's look at how we can see the samples based on this gene. Okay, here we just need to select the labels. Okay, so these labels right here. Okay, again, now we already don't care really so much about the luminal. What we care about is the separation between the rest of them. And the separation, for example, shows us, okay, so we can see that the difference between cloud and low and basal is less than 5.2. Again, that means that just like here, this was the rest was below 4.25. This, everything below 5.2 is going to be cloud and low. Above is going to be basal. Okay, so let's take, so cloud and low, you can see are the lower ones, and the basal are the high ones. You see that? So that was the separation that we see based on this gene. So a combination of two genes gave us a clear separation between three different classes of samples. And what is interesting about that is that it was done automatically, right? We didn't have to look for these genes. We didn't have to define any threshold. It gave us the genes and it gave us the threshold. In our next session, we'll talk about how the same idea can be applied to analyze large quantities of data and come up with a number of features that together gives us a very nice separation between different classes, and how we can apply this to do predictive modeling, okay? When we can get some kind of a new data set, not the same one that we use for training, and get the result, get some kind of information just like we looked at here. And now, of course, we can interpret this. We can look up, okay, well, what is this gene? And we can try and see what does it have to do with, for example, luminal or other types of data. Okay, so you can see that this is okay. So to the question that Pratim is asking, so have we created a classifier for luminal and cloud and low? Right now, this is one of the genes that has some predictive power. A classifier typically means a collection of genes, okay, not one or two, but maybe 15 or 20 or 50 or something like that. And that classifier will work for different types of data. Okay, so we'll talk about that later on, but this is indeed a beginning of that idea of building a classifier, which is the major component of feature selection when we do classification. Okay, any other questions on what we covered today? Okay, if there are no questions, I do want to mention that Mohit and Pratim will be doing a workshop at Amity University. Pratim or Mohit, do you want to talk a little bit about that, when it's going to happen, how to get in touch with you, etc.?
Yeah, hi everyone. So we are planning to do a workshop and hands-on and answer all the questions that you have. And also there are some issues with systems and something like that. So we'll be present on 24th of April and 25th there in Amity, Kolkata, and we'll be answering to all the questions and, you know, all the support that you would require.
Okay, thank you very much, Mohit, Pratim, Sahil. If you have any more questions, again, like last time, you can go ahead and send them to us by email or on chat, and we will address all of your questions and put them up. We are planning to transition to individual projects, and so in one of our upcoming sessions, Justin, who is on our team, is going to be talking about different types of applications, different types of projects, how to find a project data set if you want to train on some public domain data set, or if you have your own data set, how to prepare the data set for analysis so that we can help you go through it and do some proof. Just came from a conference, a 2-day conference, where 4 different posters were presented from our previous program at the Louisiana Biomedical Research Network, their annual conference. So we had 4 posters from there. And of course, we are expecting that from this group, a lot of you will have some interesting ideas that we can work through using different types of next generation sequencing data. And as a result of that work on your own data, you will also see kind of what are the challenges, what are the opportunities, how to put all of these different types of methods together so that we can get some biologically interpretable results. Again, thank you for joining this session and looking forward to seeing you again soon.